We left off with a brief overview of the anatomy, the major parts of the kidneys that allow it to function the way it does. And the function is what we're focusing on in class this week. The anatomy was outlined in that video nicely, and it introduced some of the physiology as well. So we're going to revisit that and follow it in order as we discuss renal physiology moving forward. But I think the major parts of the nephron, the major parts of the kidneys, the corpuscle, of course, the glomerulus, the capsule, the major site of glomerular filtration, which is why I'm bringing this back up today. And you saw the nice little animation of the podocyte, the podocyte in that video, the octopus-like cell with the little feet, the pedicles or the pedicels, whatever you want to pronounce it. The filtration slits that prevent a lot of large particles from passing through. And what was interesting in the video is they mentioned these are negatively charged, which repels proteins in the blood. Proteins, the carboxyl group has a negative charge on it, and like charges repel. So these being negatively charged help to keep larger proteins out of the filtrate. Help to keep larger proteins in the blood and not be expelled into the tubule. So you know these layers, you've seen the basement membrane, you know the fenestrations, you know the podocytes from lab. I just want to remind you of that as we get into our discussion of filtration today. So we are looking at what? Major elements of the kidney. The corpuscle, where fluid is excreted, or where fluid is filtered. This is what is responsible for the filtration of fluid. All but the largest molecules pass out. The tubule is out of the body. This is just a recap. This is not in the slides. I see some blank faces. Just a recap. Make sure we're all on the same page. And a large role of the kidneys regulating volume constituents in the blood. These are the structures that accomplish that. The tubule, proximal and distal convoluting tubules, and the loop. This is outside of the body. These carry wastes to the bladder unless substances are reabsorbed. So for all intents and purposes, things in the tubules are destined to be expelled unless they're reabsorbed. And if anything is reabsorbed, we don't know what that is yet, but we'll see that in the coming lectures. They are reabsorbed immediately into the capillaries that are in close association with those tubules. Peritubular capillaries. And those join the venous system. They send blood back to the renal veins, the inferior vena cava, and back to the heart for circulation throughout the body. So if things are reabsorbed from the nephron tubules, they're reabsorbed into the peritubular capillaries. And you can see that on the inset figure in the background of this slide. No major surprises here, just recapping the major structures as we know them. One other area that I want to emphasize, though, that wasn't, it was mentioned in the video, but sort of in passing, is a regulatory area that we'll refer to a number of times over the coming week. The Juxtaglomerular Apparatus. Juxta means beside. Juxtapose, beside something. Glomerular, beside the glomerulus. And the apparatus is the name for a series of cells in combination. It's not one definite structure like the capsule is, or like a tubule is. It's a series or aggregation of cells. And it's a really unique place because it connects the functional end of the nephron to the start. The ending and the beginning of the nephron are connected at the juxtaglomerular apparatus. It's a really interesting orientation because it essentially allows the nephron to evaluate how good a job it's doing. and regulation will proceed accordingly. We'll talk about that today. So the ascending loop contacts the afferent arteriole at the macula densa. And you can see that here. The entire apparatus is highlighted in blue. So the afferent, the arriving arteriole, contacts the distal convoluted tubule at the macula densa. So these are the cells of the tubule and the juxtaglomerular cells in the arterial wall make up the juxtaglomerular apparatus. So fluid or wastes are leaving the tubule here. They're being expelled to the ureters. Blood is coming in to the glomerulus, to the afferent arriving arteriole, and these two can communicate. The juxtaglomerular apparatus allows that communication. Mainly, it will regulate the filtering ability of the nephron by monitoring sodium, potassium, and chloride concentrations. But it importantly allows linking of the start of the nephron with the end of the nephron, and that ability to communicate and evaluate the effectiveness of the reabsorption is what's important. And we'll talk about some of the reflexes that are carried out here. Constriction, dilation, different signals, etc. But just note, this is the point of connection between the start and the end of the nephron. So the juxtaglomerular apparatus is this point between those two structures. Now, knowing that that exists, we don't know how it works yet, but we will talk about it when we get into the physiology of it. Physiology, how we filter fluid, filter ions from the kidneys. So renal physiology, the process of making urine is really secondary. The process of making urine is secondary. The kidneys are trying to maintain homeostatic pH, homeostatic volume, homeostatic ion concentrations, and to do that, urine needs to be created. It's really secondary to maintaining homeostasis. Urine formation as a proxy of kidney function is outlined here. So this is a simplified version of the nephron where you have a C-shaped corpuscle, the glomerulus with the incoming blood, and a linear series of capillaries and tubes branching out towards the right-hand side, showing you reabsorption and showing you secretion, which are the main processes outlined in the video. And so in the process of urine formation, filtration is what we're talking about today, and it's the ticket to the show. This is to be involved in regulating fluid and ion concentrations. They have to be filtered first. So filtration describes the process of expelling ions and fluid from the body, from the blood specifically, into the capsule to be excreted by the rest of the body. And Matt already highlighted to you that a lot of fluid and ions get filtered out. 99% are reabsorbed back into the body, so very little actually gets excreted, on the order of maybe 1 or 2 liters per day, but that's it, which is relatively not very much when we consider the filtration rate that I'll bring up shortly. Reabsorption is the process that helps to restore or return any of those filtered substances to the body. 99% of the filtered substances are restored, and anything else is left in the tubule to be excreted. Some things are added to the tubule to be excreted. Some different drugs are added to the tubule to be excreted. Some ions are pumped out of the blood into the tubule to be excreted. And we'll talk about those later on in the week. But the 3 major processes outlining renal function are shown here. And so if you combine all of them, whether you will excrete a substance or not is the net sum of these factors. If it's filtered, if it's reabsorbed, or if it's secreted. To excrete a solute, you would add the filtration rate, whatever is pushed out at the corpuscle, plus anything that is secreted in step 3. If it's added into the tubule, that solute will be excreted, and then anything that's reabsorbed takes away from that total. Simple mathematical equation. And we can do this for various ions if we know their concentrations at different points along the chain. So whether a solute is excreted depends if it's filtered, if it's increased, if it's secreted, and then any reabsorption will limit the likelihood of excretion of that solute. So we are focusing on the filtering part today. We'll discuss reabsorption and secretion in depth over the next 2 lectures. They are relatively complicated topics. This one is a nice introduction. It's not so complicated, but there are some concepts to wrap your head around. Glomerular filtration, and I'm always going to trip over that word. It's one that my mouth can't pronounce. Not unlike filtering water through coffee grounds, but less appetizing, I suppose. So whether or not something is filtered depends on the gradient. Here, pressure. Depends on blood pressure in the glomerulus. If pressure is high, filtration will increase. If pressure is low, filtration will decrease. That should make sense. Pressure is proportional to filtration. Now, in the corpuscle, pressure is the main driving factor of whether a substance is filtered or not, but there are other contributing factors that determine net filtration. And they are listed simply here. 2 factors oppose filtration. I'll show these to you on one of the coming slides. But capsular hydrostatic pressure describes the pressure of anything already in the capsule. You're trying to push fluid into a space and solute into a space. If there's stuff in that space, it's hard to push more in. So capsular hydrostatic pressure opposes filtration, and blood colloid osmotic pressure is the pressure due to proteins that stay within the blood. Proteins exert an osmotic pull. They don't want to let go of fluid and solids, and they try to pull and retain fluid in the blood. So those 2 things oppose filtration. But if pressure is large enough, those 2 factors will be overcome and filtration will occur. And the spoiler is, filtration always occurs, and it's very tightly regulated. So those are always overridden by pressure within the glomerulus. And when pressure is higher than those other 2, water and ions move out. We'll call them by name eventually, but right now, water and ions, water and solutes, small molecules, glucose and the like, move out. They're overridden to such an extent that 150 to 180 liters per day leave the blood through this mechanism. 150 to 180 liters per day leave through this mechanism. Only 1 to 2 liters are expelled completely. So 148 to 178 liters are reabsorbed. Very little protein because of the small slits and those negative charges that repel proteins. Just small molecules, urea, glucose, and ions, and fluid. If you, I'll bring this up later. So lots of filtration going on. 150 to 180 liters means a lot of filtration going on. Let's consider the elements that make the glomerulus the ideal site for filtration. And I threw this image together to help get across 3 main points that make this the ideal site for filtration. It's a pretty rudimentary schematic showing what I hope is obviously the afferent and efferent arterioles. You have the glomerulus, which is meant to be coiled, the glomerular capsule. Both of these things are the corpuscle, and then the tubule as it extends outwards. Proximal initially here, loop, distal, ureter, so on and so forth. But this is the site of filtration. In understanding filtration, it's really the same process as capillary exchange. We talked about capillary exchange at length with gas exchange or the delivery of glucose to the muscle. This is the same exact process. Filtration is capillary exchange. Same concept, but a slightly different organization. And this different organization makes the filtration or the net movement of substances greater here than at the capillaries. But it's the same principle. So I want to use this slide to illustrate why filtration is greater here than at the capillaries. So in this sense, filtration is the use of pressure to force fluid or solutes through a membrane. If I contrast that with the capillaries from earlier in the semester, we used the pressure gradients for oxygen, or CO2, to force substances through a membrane. Same principle. Filtration here is the use of pressure to force fluid and solutes through a membrane. Instead of PO2, we're actually using barometric pressure in this case, but same principle. Total filtration or movement is greater here than at standard capillaries that we know and have experience with for three reasons. Compared to normal capillaries, the glomerulus has a larger surface area. Right? So this white highlighted area shows the surface area for filtration. And it's larger here than at a standard capillary because of all the convolutions and loops. Larger surface area. There is a large surface area at capillaries. They branch and make a meshwork, but they don't coil and loop like the glomerulus does. Two, this membrane is very thin. Even though you have three layers, podocytes, the basement membrane, even though you have three layers, it's very thin and thinner than the capillary membrane at the tissue. So this allows more filtration to occur. And the pressure gradient is bigger in the glomerulus, pushing more fluid and solute out. And so what this horizontal line is meant to represent is the bottleneck created at the glomerulus. The efferent arteriole is a little bit smaller than the afferent arteriole, which creates a bottleneck and increases pressure in this area because more blood arrives than can leave the structure. So pressure is increased due to the structural differences in the arterioles. We have a large surface area, a thin membrane, and a high pressure gradient. You might think these elements are familiar, and they are the exact three things that we talked about that made the lungs the ideal site for gas exchange into the blood. High surface area, thin membrane, and a pressure gradient. These are things that are different than the standard capillary beds that we've discussed before. And so filtration here is larger than that initial concept that we have of the exchange of solutes or ions across a membrane. So let's look then at the tendency for movement. Let's look at pressure a bit more closely. There's a high pressure here created by the difference in the size of the arterioles, and we naturally regulate blood pressure at a slightly higher value here to achieve that gradient. Where it's really, really low in the capillaries, near tissues in the body, pressure in the glomerulus is about 55 millimeters of mercury. So pressure in the glomerulus on the arterial side within these arterioles is about 55 millimeters of mercury. That's high. If you think back to, I showed a schematic on the vasculature side showing pressure change as you move from the aorta to the arterioles to the capillaries to the veins, and it is a linear decrease over time because blood flowed from high to low pressures. 55 millimeters of mercury is fairly high for a pressure in the glomerulus, but this is what drives filtration. This is point number one. This red arrow pointing out, this is the driving pressure of fluid and solutes leaving the blood because of this high starting pressure. Now, the things that oppose that movement were listed earlier and are shown here. There's residual pressure inside the capsule. You're trying to push fluid and solutes into a space that is already full. And so there's some pressure that resists that movement. It's not a lot of pressure, but there is some pressure. If we were to put a number to it, it would be about 15 millimeters of mercury. And that is highlighted here with this purple arrow. The capsular hydrostatic pressure that opposes the new injection of fluid or small molecules. Also opposing filtration is the colloid pressure of proteins that are kept within the blood. They create a pull that wants to prevent fluid from leaving. Prevent fluid and small molecules from leaving. So that the proteins are not filtered out and stay in the arterial blood, the pull exists that tries to restore fluid that had been pushed out on the order of about 30 millimeters of mercury. It's a fairly large equivalent pressure. Now, these are generally pretty stable. The thing that we have the most control over is the initial filtration pressure, the initial blood pressure in the glomerulus. We can increase that, we can decrease that, depending on whether we need filtration to go up or down. We have a lot of ability to regulate pressure to push fluid out and to filter small molecules into the capsule. Combine all those things together and you get a net filtration pressure of only about 10 millimeters of mercury. That says there is a gradient. From high to low, fluid and small molecules move out of the arterioles and into the capsule because this number is positive. The opposition is not big enough to override pressure in the glomerulus. Hypothetically, if net filtration pressure were zero, there would be no filtration. That never happens, but hypothetically. Also hypothetically, if net filtration pressure were negative, fluid and solutes would move into the blood. Again, that never happens here, but you can understand what I'm trying to illustrate. It's about 10 millimeters of mercury, and we can change that number. We can change the rate of filtration by modifying blood pressure in the arterioles. So what we're talking about today is how we modify that pressure. So Pressure is actively controlled to maintain filtration rate. We are always regulating afferent arteriolar pressure to maintain filtration rate. And filtration rate would be the amount of fluid in all the nephrons in both kidneys being filtered out of the blood per minute. The amount of filtrate formed by both kidneys and all the nephrons in all the kidneys per minute. And we need this value to stay relatively constant. We don't want it to fluctuate wildly. We don't want it to hit zero. In the face of low blood pressure, we want it to go up so that it's not compromised. If you have chronically high blood pressure, you need it to go down so that we don't expel too much fluid or too much small molecules and ions. It's tightly regulated and usually quite constant. And so we're talking about how we can keep that filtration rate constant. Okay. So, what is constant? What filtration rate is normal? What filtration rate is constant? Let's give some context. All of the nephrons in both kidneys will tend to filter out 2 mils per second. 125 milliliters per minute in men, 105 milliliters per minute in women. That amounts to about 2 milliliters every second leaving the blood and entering the tubules of the kidneys. Every second, 2 milliliters. Right now, 6, 8, 10, 2 milliliters every second. Over the course of your 50-minute lecture, 6 liters will be filtered out. It's a pretty high filtration rate. And that is maintained tightly by renal autoregulation, neural regulation, and hormonal regulation. Now, these are somewhat tiered. Renal autoregulation is quick, immediate responses that maintain glomerular filtration rate within a tight range and keep it stable. Neural regulation can override autoregulation in certain situations, and hormonal is more chronic or long-term. We'll look at those coming up. Now I'm going to replay a quick portion of the video for you, but I want you to understand that filtration rate is proportional to net filtration pressure, or it's proportional to blood pressure. That's the thing we're regulating, okay? Now we've seen this already, but I want to remind you of it.
The glomerular filtration rate, or GFR, is the speed at which filtrate passes through the glomerular capillaries into the glomerular capsule. GFR is controlled by a process called renal autoregulation and contributes to maintenance of plasma concentration and removal of waste. If the GFR is too slow, too much is reabsorbed and plasma becomes too concentrated. Excessively high GFR doesn't allow time to reabsorb sufficient solutes back into the bloodstream.
Okay? So you can understand each of those. Before I put that up, if GFR is too slow, there's not enough filtration. Very little fluid, very few substances are leaving the blood. The blood becomes too concentrated. All right, filtration is too slow. Nothing is excreted from the body. On the other hand, if filtration is too high, large amounts of fluid are excreted, large volumes of ions are excreted, large amounts of glucose are excreted. And the kidneys only have a certain capacity to reabsorb those things. So if GFR is too high, a lot of those will be filtered out and expelled, more than we want. So we need to maintain a stable GFR. And we have to be able to respond in a case where it's too high and bring it back down, and vice versa. And we do that by affecting afferent arteriolar flow, or afferent arteriolar pressure. We regulate on the afferent side. So we've seen the three things that will result in filtration. And pressure in the glomerulus is by far the most important. So we directly regulate pressure in the glomerulus by modifying afferent blood flow. If afferent blood flow is high, so a lot of blood is coming in, pressure will be high in the glomerulus. GFR will be high. Blood flow is low, little blood is coming in, pressure in the glomerulus is low. So we acutely regulate the afferent blood flow in order to dictate glomerular filtration rate. How do we regulate afferent blood flow? This is the process that is controlled by renal autoregulation. And there's two branches of that autoregulation. And autoregulation simply means automatic, instantaneous automatic regulation. It responds moment by moment to varying stimuli. So the first branch, the myogenic mechanism, relies on stretch of the afferent arterioles. A lot like baroreceptors, detected stretch and regulated blood pressure in the carotid arteries or the arch of the aorta. There are stretch sensors in the afferent arterioles. So, if blood flow in is high, if pressure is high, stretch in the afferent arterioles is going to be registered. It'll be firing at a high rate. When that happens, the myogenic mechanism dictates there is a reflex contraction to close off the afferent arteriole, which limits how much blood can come in. What that accomplishes is there's less blood arriving at the glomerulus, pressure in the glomerulus drops, and so filtration drops back down to normal. Think of what would happen in a low blood pressure situation. And I'll leave that to you to think of on your own. We also have, so this is a feed-forward type mechanism, right? We have stretch receptors in the afferent arterioles that we can regulate the incoming blood flow on a moment-by-moment basis. We also have feedback. The second branch of autoregulation is feedback. Feedback. We can measure and see how well substances have been filtered out. If there's too much of them being excreted to the bladder, we will adjust the glomerular pressure accordingly. And this mechanism describes the tubuloglomerular feedback. This is executed through the Juxtaglomerular Apparatus that we introduced at the start of class. We will look at the constituents leaving the nephron and see whether or not too much solute is being excreted. So in a situation where we have high blood flow in and high filtration, we have a high GFR, lots of stuff is being filtered out, expelled into the tubules. Lots of fluid, lots of ions, lots of small molecules. The kidney can't reabsorb large volumes of these solutes if they've been expelled. Or if they've been expelled in large volumes, the kidney has a certain capacity with how much it can reabsorb. So if GFR is very high, there's a lot to reabsorb. Not all of the solutes can be reabsorbed, and that is sensed on exit from the nephron by cells in the macula densa. They'll monitor the composition of the excreted fluids. If it's too concentrated, too salty, if there's too much in there, it will regulate the afferent arteriolar blood flow to reduce GFR. So if there's many substances in the filtered blood, the kidney can't absorb them all. The macula densa will inhibit the release of nitric oxide. Now, nitric oxide we've seen before, very potent vasodilator. This was the thing that the muscles used to say, send blood here during exercise. Nitric oxide is the thing in pre-workout drinks. They give you a really nice pump, or it makes you feel really veiny. Vasodilator. Very potent vasodilator. So, if too much is being excreted and we inhibit vasodilation, that means there's less dilation, therefore the arterioles constrict, they get smaller. Less blood ends up in the glomerulus, less GFR ensues, less stuff is filtered out. So I'm going to recap that mechanism again really quickly.
A constant GFR requires a stable glomerular pressure. The myogenic mechanism contributes to this stability. When increased blood pressure causes renal arteriolar smooth muscle to stretch, automatic constriction of this smooth muscle will result because of this mechanism. When systemic blood pressure is low, the same muscle relaxes to maintain a constant GFR. GFR can also be adjusted by tubuloglomerular feedback. Within the wall of the distal convoluted tubule is the macula densa. These cells detect the flow of filtrate through the nephron by monitoring sodium and chloride ions and water passing by. The cells secrete paracrines to adjust the diameter of the afferent arteriole and control glomerular pressure. If the flow is too slow, the afferent arteriole relaxes, increasing pressure in the glomerulus and increasing GFR and flow through the nephron.
Right? So the flow is too slow, filtration is too slow, not enough is being pushed out. We open up the afferent arterioles and allow more blood in, increasing pressure, increasing GFR. You have this as a summary in your book that looks like this, the typical negative feedback loop. And I zoomed in here to show you the Juxtaglomerular Apparatus and how we have the distal convoluted tubule in very close proximity to the afferent arteriole. And so after all this work has been done to reabsorb solutes, we can communicate how well that has been executed. How well has the nephron done its job of reabsorbing solutes? And then if we need to adjust, we can change the afferent blood flow accordingly. We're essentially measuring the effectiveness of that reabsorption and adjusting flow if needed. That was the tubuloglomerular feedback, the second branch of autoregulation that we just talked about. Now let's just briefly go over the two other mechanisms of regulation. This is primary. This is what you should know if you know nothing else, renal autoregulation. The other mechanisms can override this if needed. Renal autoregulation by far is responsible for the majority of the stable GFR at rest, and it can be overridden in non-rest situations. So we have the ability through neural regulation to override the local regulation by the nephron proper. So we have a large sympathetic innervation of the kidneys. We can constrict both the afferent and efferent arterioles equally if we need to. We don't do that very often. At rest sitting here, there's no sympathetic drive. You're not in a fight or flight situation, and you're not constricting those arterioles via this mechanism. Right now, for you sitting here, autoregulation reigns supreme. But if you were to jump up and run out of class, like whoever that fellow was just did, jump up and run out of class, start to exercise, you engage the sympathetic system, and you can override local autoregulation. What benefit does this have? Why would you want to override autoregulation? The sympathetic system will clamp both arterioles, not shut, but really close them off. And this does two main things. If you're exercising, you probably don't want to take a lot of breaks. You don't need to make a lot of urine. And so it decreases filtration and decreases urine production. Really good for marathon performances or Ironman triathlons. Sympathetic activation also makes sure blood is available to the tissues that need it. In that situation, the metabolic implication is the muscle needs to contract, you need to do work, maybe to survive, who knows? But you don't need to worry about pH, volume, ions acutely on the short term. Blood needs to be diverted to other organs and other tissues to accomplish whatever goal is required at that point in time. So we can override autoregulation by engaging the fight or flight branch of the sympathetic nervous system. Both of these are relatively acute. Autoregulation, moment by moment basis. You'll never really see an appreciable difference in GFR. Neural regulation will change GFR drastically, quickly. Hormonal regulation can change GFR, but on a more prolonged basis. Very similar pattern to how we've seen neural and hormonal regulation layered in the past. Neural is quick, signals are really fast. Hormonal regulation takes a bit longer because you need to secrete and activate hormones in order for there to be an effect. Now we'll come back to angiotensin II. We've seen this already. I highlighted the cascade at the beginning of this series of slides, and we saw it in the blood pressure section. But angiotensin II, vasoconstrictor, constricts the afferent and efferent arterioles if we need to decrease GFR. This would happen in a situation where you've lost a lot of blood or you're hemorrhaging. You want really chronic, large constriction. You don't want to get rid of a lot of fluid. You don't have a lot of fluid to give. And so angiotensin II is a hormone that will help prevent the extra loss of fluid in a situation where you might be hemorrhaging. On the flip side, maybe you're not hemorrhaging, maybe you have chronic high blood pressure, and hormonal regulation needs to increase GFR to help get rid of some extra fluid and decrease pressure in the body. Candidate hormone that does that is called atrial natriuretic peptide. Atrial natriuretic peptide. And ANP is secreted from cells in the heart, secreted from the atria, atrial natriuretic peptide. When the muscle cells of the heart stretch, when there's a lot of blood, there's a lot of volume, indicating lots of pressure, this is released, and its job is to increase the surface area for filtration, which isn't a way that we regulate filtration normally, to change the surface area for filtration. And it does that by changing, let's go back, there we go. By relaxing the mesangial cells, which are in between the afferent and the efferent arterioles. This is considered to be the pole, the flagpole of the glomerulus. And when these are contracted, through mechanisms that I don't fully understand, the surface area is reduced. When they are relaxed, the surface area for filtration increases. So atrial natriuretic peptide, when there is high blood pressure, lots of fluid on hand, it's released from the heart, it relaxes these cells, increases the surface area so that more fluid can be expelled from the blood, hopefully decreasing pressure and restoring homeostasis. So we're going to leave it there for today. That's filtration. Make sure you know autoregulation. That's important. We're coming back to reabsorption and secretion on Thursday and Friday. Have a wonderful Tuesday afternoon.